for the real marathon start. We have only done 1 kilometer. There are 41 remaining. So, so as Otto introduced me already, I'm Nerea Abrego. I am from the University of Helsinki, but now transitioning to the University of Jyväskylä. And in this lecture, I will introduce community ecology very briefly. And I will also talk about the typical data that is collected in community ecology. I will introduce community ecology with the more specific aim of linking it to HMSC, so that we ask or we answer the question of how does HMSC relate to theory in community ecology. I will also talk about the typical data that is collected in community ecology, but also with the specific aim that I show you, or I explain what kind of data HMSC takes as input. So this lecture is general because we are going to introduce community ecology in general, and we are going to talk about data in general, but is specific because is targeted to or is linked specifically to HMSC. So before we go into real theory in community ecology, the first question that we should answer is, what is community ecology? So community ecology is a multidisciplinary field that aims to describe and understand the spatio-temporal structure and dynamics of ecological communities. However, community ecology as a field, it has relatively only recently rooted in ecology as a field, and its entity as a field, it has been strongly questioned. In fact, one of the most influential papers in community ecology is that one by Lawton in 1999 that heavily questions community ecology as a field. He said about community ecology is a mess with so much contingency that useful generalizations are hard to find, meaning that we have in community ecology so many study system specific studies that it's difficult to make generalizations from all of these individual studies. But since then, the last 2 decades have experienced a proliferation of unifying theory and general conceptual frameworks in community ecology, out of which I will explain the most renowned ones. Okay, so now we have answered the question about what is community ecology, but then what is an ecological community? So in the same way as community ecology as a field, it has been shaping only recently the term ecological community, it has also acquired disparate meanings through history. But nowadays, the most broad definition that we accept or that we use is the assemblage of at least 2 potentially interacting species at a given time and location. In typical HMSC analysis, we usually work on data on specific taxonomical groups, either because we have been trained to identify specific taxonomical groups, or because the methods that we are using to identify the species, they are taxon specific. And often it is unclear in our data how the species are ecologically related, meaning that, for example, if we are working with atlas data, where the data have been compiled at different time scales or the spatial scales are very broad, conceptually, it's difficult to know a priori or we don't even know whether the species that we have, they are ecologically related. In which case it might be a more proper term to use species assemblage data. But because of these issues, a question that often is raised by, most likely is a question that you have got from reviewers in your research papers or by colleagues, is can we really use the term ecological community with our data? Well, as I already mentioned, there are plenty of definitions of ecological community, and some researchers like to stick more to some more strict definitions, and some others to more flexible definitions. But in HMSC, we follow the consensual definition by Both and others that he published in 2016 in American Naturalist. And that definition of ecological communities that we use in HMSC is a collection of species occurring in the same place at the same time, the species not being necessarily restricted by phylogeny or resource use, and allowing the spatial boundaries to be either natural, for example, if our sampling units are islands or resource units or ponds, or arbitrary, for example, if we have study plots or study sites. So this is the consensus that we follow in the HMSC book, and that we will also follow through this course. So after introducing these very, very basic concepts of what ecological communities are and what community ecology is, it's time to go a bit more into ecological theory. While it's true that there are like plenty of conceptual frameworks in community ecology, there are three frameworks or three general theories that are nowadays the most renowned ones, in the sense that when we look at studies in empirical community ecology, they are the ones that we use the most to frame our study system into theory. These are the assembly rules framework, the meta-community theory, and the most recent balance theory of ecological communities. HMSC is most closely related conceptually to the assembly rules framework, as I will explain in the next slides. However, as I will also explain, we can also relate our HMSC results to meta-community theory and to balance theory. So what is the assembly rules framework about? Well, the term assembly rule, it was first introduced by Diamond in 1975 to refer to the restricted species combinations to which competitive interactions can lead. In his famous work in the New Guinean archipelago, Diamond found that in some islands we find one species of these fruit dogs or the other species, but we hardly find two species combinations into the same island. And in his view, this was because of competition. So in principle, the term assembly rule, it was used to refer to competitive interactions only. However, after Diamond's work, the meaning of assembly rule has broadened from competition to any ecological process favoring or disfavoring the occurrence of a species. And more generally than in Diamond's definition, co-occurrence of a species, co-occurrence or occurrence of species is a product of stochasticity, historical patterns of speciation and migration, dispersal, abiotic environmental factors, and biotic interactions, with none of these processes being mutually exclusive, meaning that all of these processes, they are working together, shaping ecological communities. And if we put all these assembly rules into one conceptual scheme or illustration, we get this kind of illustration that I just put here, which you might have already seen if you have been following the HMSC publications, but also very similar conceptual illustrations are used elsewhere. Also, at this point, let me make the point that rather than assembly rules, nowadays we use more the term, or we talk more about assembly processes. This is because if we use the term assembly rules, the problem is that rules is a very strict term, and we know that in ecology there are very few rules. If we use the term assembly processes, this term is more flexible, and it also allows to consider the more dynamic that processes are more dynamic than rules. Well, so if we try to understand this illustration that is in the left-hand side of the slide, let's start by the very general concept, or the very general concept that the assembly processes or assembly rules can be conceptually viewed as filters that act at the scales ranging from the regional species pool, from the larger spatial scales, to increasingly thinner scales until the local community composition. And what are these processes or rules? Well, the first category is the phylogeographic assembly rules, which refer to the historical patterns of speciation and large-scale migration. The ecological assembly rules, which might be the most familiar to you, refer to the smaller scale dispersal, which are also known as dispersal assembly rules, the effects of the abiotic environment, which is also known in the ecological literature like environmental filtering, biotic interactions, which we often refer to as biotic filtering, and stochastic processes like demographic stochasticity, for example. And a very important concept or part of the assembly rules framework are actually species traits, because many species traits can influence the ecological assembly processes. These sort of traits, these sort of species traits, they are known as response traits. So, for example, the competitive ability of a species will obviously affect biotic filtering. The dispersal capability of a species will obviously affect the dispersal of a species. and the resource specialization level of a species in a community will affect the effects of environmental filtering. So we often try to detect in community ecology these species traits. Is an important part of in empirical community ecology. So how does HMSC relate to all this stuff that I just explained? In 2017, we published this paper in Ecology Letters, which, by the way, is open access, and you can download it from our webpage, where we explained how the different HMSC components could be related to the different assembly processes or assembly rules in the assembly rules framework. In this figure here, I have, if you will realize that I have already put some parameters here, because we have not yet introduced the HMSC model. I am not going to go deeply explaining what each of these parameters or outputs are, but I thought that it is already good to have them here in a figure that relates to ecological theory, so that later on you will go assimilating things or the contents of the course better. So while HMSC is not a process-based model, modeling framework is a correlative statistical method. Let me remind you that with HMSC, we are not directly modeling these assembly processes, but the different components of HMSC, we can relate them to these assembly processes so that we can relate the different HMSC components to theory. For example, environmental filtering, we capture it or we look at it through the so-called beta parameters that measure how the species respond to the environment. Biotic filtering, we can assess it through the so-called omega matrices that measure at which level and how the species are associated among them in our species community data. We can measure the influence of traits through the so-called gamma parameters. The gamma parameters, they measure how the traits that we have included in the analysis influence the responses of the species to the environmental conditions or to the environmental covariates that we have included in the model. The so-called raw parameter relates to the relatedness, to the phylogenetic relatedness of the species, and it tells us about whether species that are phylogenetically related respond more similarly or less similarly than we would expect by random. We can also, with HMSC, relate our results to dispersal if we, for example, have spatial data, as the gamma parameter, or alpha, sorry, no gamma, alpha. The alpha parameter will tell us whether there are spatial structures remaining in the data that we cannot explain by the environmental conditions, and we may therefore link these to dispersal processes being acting in our community. And finally, because with HMSC is a model-based approach, we can calculate the predictive power of our model, which means that we can also measure how much variation is not explained, and we may relate this to stochastic processes being important actors in our community. So this might now sound a bit heavy, but this will become hopefully clearer and clearer as the course advances. So I think that is now time for a short break. So let's meet again 1:15, so more or less in 5 minutes from now, and I will continue with meta community theory, and maybe Mirka will tell me if there were any questions in the lecture so far.
Nerea, we have a single question. Maybe you want to look at it during the break and.
Okay, yeah, let's do that. Okay.
Sorry, what time are we continuing?
1:15 or maybe 16 past.
Okay, in 5 minutes.
Yeah, 5 minutes break.
Great. Okay. Thanks.
See you. So here we are. Somebody was asking about whether we can include genetic variation in the analysis. Well, HMSC at the moment focuses on species level data. So if you, for example, have genetic variation data at the species level, you can include this as a trait. But if you have population level genetic variation as your data input, then HMSC is not yet developed for this. And actually, an extension for HMSC that is pending and that we have already started to think about is how to add one more layer of variation at the, not the species level, but the individual level, so that we could add this kind of components into HMSC. I just saw another question, which was about what happens if different processes overlap in creating variation in our data. Well, then this is something that is actually common that it happens, because many processes are acting at the same time, and different processes may create similar patterns in our data, and HMSC just picks up these patterns in the variation of the data, and as any other statistical method. So if you suspect that this might actually be the case with your data, this you need to deal with in your own study system and think about it a priori before making any deep conclusions from the HMSC outputs, but the same applies to any other statistical or relative statistical method. So if you are ready, I will share my screen again and we continue with that.
Sorry, Nerea, actually.
Oh yeah, sorry.
There was still one more question. I don't know if this is something that Otso will return to, but it was about, so the question was, are dispersal patterns related to latent or random factors since they are associated with the data spatial structure? But I guess this is something we're going to be diving into.
Yeah. But as a short answer, let me answer that this alpha parameter is in fact estimated from the random spatial from the spatial latent factors. So yeah, but with Otso you will go like step by step, really understanding where each of these parameters come from in the model and understanding more like how they are related and hopefully also interpreting them. Now, my aim was just to introduce them for the first time, to show you that they are related to ecological theory, because that is our ultimate aim when we apply any analysis. So if we are ready, I share my screen. So I'm pretty sure that for most of you, this meta-community theory is one of the theories that sounds strongest to your ear when we think about community ecology theory. In fact, this has been one of the most influential theories or frameworks to understand empirical community ecologies, empirical communities. So meta-community theory explains how networks of local communities result from the interplay of stochastic, like demographic stochasticity, and deterministic, like niche differentiation, competition, and dispersal rate, processes acting at different spatial scales. So from this illustration from Labeled and Chase's book, you can see that in meta-community theory, we have local communities, well-defined local communities, which are represented by circles, which are linked to other local communities by dispersal, which is represented by arrows, the thickness of the arrows representing the rate of dispersal. And meta-community theory synthesizes 4 perspectives, which are also known archetypes or paradigms, which each arises from a different but not mutually exclusive conceptual perspectives. These are the neutral paradigm, the patch dynamics, species sorting, and mass effects. And I'm not going to explain them one by one, because that would be then I would need to give a course in community ecology rather than a course in HMSC. But the reason why I have put this here is that quite often when we apply HMSC or when we are analyzing any community data, we are trying to find out to which of these paradigms our community mostly is linked to. This is a very common aim when we are in community ecology, when analyzing community data. However, if we look at empirical studies trying to figure out which paradigm from the meta-community theory the data mostly fits to, it has been proven to be quite difficult to really disentangle the different paradigms from empirical data. And there are different reasons for this, but perhaps the most important reason for this is that the local communities, they are often more continuous than they are assumed to be in meta-community theory. If we look at the left-hand side of my screen, we see how in meta-community theory, the different habitat patches where the local communities are organized. In meta-community theory, they are supposed to be organized in very well-defined spatially defined local communities. However, when we go to the field and we try to sample our communities, we are rather in this kind of situation. And this is just because in reality, habitats they are more continuous than in theory. So usually our sampling units, they rather belong to this kind of, they belong to this situation. Therefore, can we link HMSC to meta-community theory? Well, of course we can, but you always need to keep in mind the nature of your data, because it usually comes from this kind of situation with non-well-defined habitat patches and local communities, and therefore it becomes more complex to really go to the processes that are behind the patterns that you find in your data. But of course, we have been ourselves interested in this question, because it's a fundamental question in community ecology to know or to link our results from our data to meta-community theory. So with Otso and Joel, quite recently we did this study in which with an agent-based model we simulated different meta-community scenarios. We simulated data that belonged to these different meta-community scenarios in meta-community theory. And we also simulated a virtual ecologist that would collect data from these meta-community scenarios using a spatial sampling design in a similar manner that we empirical ecologists we do in reality. And on top of collecting the data, this virtual ecologist also analyzed the data. We put this virtual ecologist to analyze the data using different statistical methods that we use in community ecology, out of which, of course, we also told her to apply HMSC. So the aim of this virtual ecologist was to try to link the patterns that she found in this data that she collected to the meta-community scenarios but we didn't tell her, of course, like what were the different scenarios that we simulated. So how well are we able to disentangle between these meta-community scenarios when we collect this kind of non-manipulative observational data and we apply different statistical methods to this data? Well, the results were perhaps a bit disappointing, and perhaps they sound a bit simplistic, but is actually quite difficult to disentangle between these among these four paradigms when we collect data in this way. The species sorting and mass effects paradigms, they create more predictable patterns in our data than do the neutral and patch dynamics paradigms. So the key message here is that if we want to look at these meta-community scenarios from our data, then it's a good idea to look at the predictive power as a statistical output. And with HMSC, we can calculate the predictive power through cross-validation, as Otso will explain in the next lectures, so that though the species sorting and mass effects scenarios, they created more predictable. We had a higher predictive power than we had with the neutral and patch dynamics scenarios. And of course, we have more predictive power when the effects, when the environment has a big effect on our species community, which we measure through these beta parameters, which are going to become your best friends in the coming days. So yeah, we can relate HMSC to meta community theory, but perhaps in a more disappointing way than you might have expected. But this is not a shortcoming of HMSC. This is a shortcoming of the data that we usually use in community ecology, and is a characteristic of any statistical method that you might use for this kind of data. And the last theoretical framework that I promised you I would introduce you is a very recent theory, which Belend proposed for the first time in 2010, this paper that I have put here. So the very big message that Belend gives us is that all the processes which I have been talking about, biotic filtering, environmental filtering, dispersal, speciation, blah, 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 we can synthesize into 4 high-level processes. So all the processes that are acting in shaping our ecological communities, we can synthesize them into just 4 high-level processes. These are speciation, dispersal, drift, and selection. So speciation creates new species through evolutionary processes. So it kind of adds species to our local community. Dispersal refers to movement of organisms in a space, and it refers both to past large-scale migrations and to contemporary small movement of species or individuals. And it usually also adds species to our local community. Drift refers to the stochastic changes in the occurrence and abundance of species through this demographic stochasticity. For example, that small populations, they go extinct and so on, and it usually removes species from our local community. What is selection? Selection are all those processes creating deterministic fitness differences among individuals of different species. So that, for example, here, the white moth, the white species, has some trait that makes it have a better fitness to the environment that it is, and it becomes more abundant, for example. That would be a selection process. So how can we relate our HMSC results to this balance theory, which has recently been proposed and is yet quite unexplored, but is so general that we want to relate to by our results. So here in the upper part of this slide, I have put this same slide that I had in the assembly rules framework on how HMSC relates to the assembly rules framework, how different parameters relate to these different assembly processes. And in the lower part, I have put balance theory. So remember that with imbalance theory, we call these processes, like these four processes, like high-level processes. And Bellen himself calls these different assembly processes that, for example, we talk about in the assembly rules framework as low-level processes. So actually, we can classify all of these low-level processes into each of these high-level processes. And here I have colored. Each of the processes in the assembly rules framework that we can relate to with our HMSC results according to which high-level process they would belong to. So environmental filtering and biotic filtering, they relate to selection. So some species, they have higher fitness in some environmental conditions than in others, which is what environmental filtering is telling us about, and this would be a selection process. The biotic filtering is also a selection process. Some species have higher competitive ability than others, and those that are stronger in competing, they remain, or so they are selective in that sense. Also, if we want to capture the effects of traits or phylogeny on how species respond to the environment, this is clearly a selection process as well. This alpha parameter that I had previously linked to dispersal, it obviously belongs to the dispersal high-level process, and this indirect way of looking at stochasticity through the predictive power that I have very briefly introduced, then it would belong to drift. So again, can we relate our HMSC results to balance theory? Yes, we can. It's quite an indirect way of doing, but this would be the way in which we would do it. So the next part of the lecture is about data. It's not anymore so much about theory, and I am sure there are some questions on my lecture so far. So I could maybe answer some of the questions before we have a very small break before the data part of the lecture. So maybe I can stop sharing and.
We've actually been answering questions in the chat simultaneously. I'm not sure if somebody had an unanswered question, maybe they'd like to paste it again quickly, but I think we have pretty much gone either answered or said that we will be returning to this.
Yeah, so
Okay, now we have one question, which is, does HMSC need true species absences? So yeah, recorded absence rather than, I guess you mean data rather than pseudo absences.
Yeah, HMSC doesn't deal with this presence only data that we may have in many databases. But also we'll explain better this issue about like when you have like true presence absence data, or you can also have the option of not having data for some of the species in the data matrix, but I think that these questions are perhaps better to be answered a bit later on when we start introducing the data and also the model, because now we are a bit like in the theory part. And and then I wanted to remind the participants that we plan to compile all these questions into one document, and we plan also to look at them later on, and to create a document where we perhaps explain the answers, explain the answers a bit better, and also if so that don't worry if we have not given like a very good answer now that we are like giving the lectures, that we try to come back to this topic later on and to publish our answers in the HMSC webpage as well. Yeah.
So will you have a short break now before continuing?
Yes. Yeah.
When shall we return?
Yeah, let's return again in 5 minutes, if that is okay.
Shall we return at 12:50 finish time? If I put it in GMT.
Okay, yeah, let's do it.
Okay.
That way. Hello. So now I will talk about a kind of quite different topic, which is what are the typical data that we community ecologists collect, and specifically I will try to answer. Wait, I didn't share the screen. Yeah, and so I will talk about what data we typically collect in community ecology, And specifically, I will try to talk more about what data HMSC takes as input. So let me start by the very general and a bit boring topic that we can, in community ecology, we can collect the data either from experimental studies or from non-manipulative field observations. So in community ecology, some of us, we work with laboratory experiments with bacteria or whichever small organisms. Some of us, we work with field experiments. So for example, if we translocate some communities here and there, and most of us, we work with non-manipulative observational data, so that we just go to the field and we collect the data on what species we find and in which kind of environmental conditions. So these non-manipulative observational data are the most common type of data in community ecology. And also, as we have seen from the data that you have submitted, in which most of it is this sort of data, in which we go to the field and we collect data on what species there are and what environmental conditions they are in. So while this sort of data is the most common, is also the most challenging to analyze. And this is because these data, they are shaped by the full complexity of the assembly processes. When we are working with data from experimental studies, like in laboratory experiments or in field experiments, we can control for certain processes which we want them to be working on or we don't want them to work on. But when we go to the field and we collect data on what the species are where, then nothing is under our control and the full complexity of processes are working on at the same time. And this is challenging because then it's very difficult to link causally the patterns that we observe in the data to the underlying processes. Still, this is our main aim when we collect the data. So what sort of data will we collect? Well, we usually collect some community data, some species data, which we accompany by environmental data, meaning that some environmental conditions which we hypothesize to be important in determining the variation in our focal community. But sometimes we also want to include some trait and phylogeny data because we hypothesize that there are some traits of the species that are influencing how the species in our community respond to the environment, or because we have some specific hypothesis in mind. And when we are collecting data, we also collect some data on the spatiotemporal context, meaning that if we go to the field at different times, then we have the data on which time we have gone to the field. And if we have like different sampling units in a space, we also have data on where those sampling units are. So let's go a bit to look at these data matrices one by one, a bit more deeply. So through the book and through the course, we talk a lot about the community data. When we are talking about the community data in this course and in the book, we refer to the data about the species occurrence or abundance in a set of temporal or spatial replicates, which we refer to as sampling units. So depending on the objectives of our study, subject organisms, we record the community data in very different ways. Sometimes we record the species directly. We go to the field and we observe the species directly and we note it. But some of the times it's difficult to do so, and we record our species through indirect cues like animal tracks. Direct species observations, they are the most common data, and for example, when we work with plants or with insects, we go to the field and we record them directly. Some other times we may use sound recorders or camera traps to record birds and frogs or mammals, and also let me note that this is a kind of direct observation, because then we will observe them from the photos that we take or from the recorders that we take. And then for some mammals which might be very shy to be observed directly, we record their presences through their indirect cues, such as tracks and other sort of droppings. But recently in community ecology, we are seeing a big rise of another kind of indirect of data coming from indirect observations. And these are the so-called DNA-based molecular identification or of environmental samples. This is also an indirect way of recording a species. So for example, if we go to the field and we collect one soil sample, and then we apply DNA barcoding to record what species of fungi, bacteria, or microarthropods are in our soil sample. This is an indirect way of recording the species. And this sort of data are becoming very common in community ecology, because they are so efficient to record many species groups that so far they were difficult to record, like if we think about microorganisms and so on. This meta-barcoding is done by amplifying and sequencing the barcode gene regions. For example, the ITS region is commonly used for fungi, and the COI region is used for animals, and so on. And here we have an illustration of how we get this data, but this is not really it's just an illustration, it's not the point to explain how this is done. Just to say that this sort of data is becoming very common, as we have also seen from the data that you have submitted to for this course. Another sort of community data that is becoming increasingly common in community ecology is data that originates from databases combining many sources, like the Global Biodiversity Information Facility is a famous source of this sort of database, and the iNaturalist citizen science platform is also a quite famous source. So, for example, in this Global Biodiversity Information Facility, if we look at how many papers, scientific papers have used this sort of data for scientific purposes, we see that these are quite significant numbers. This sort of data, they are becoming also very common. And finally, just a very quick reminder that which you may not need, because this is very basic, but still I want to remind, is that we can measure the species in many ways. We can record their presence, or we can record presence absence, or we can record their abundance, which we can do in many ways. Like, for example, in plant communities, the abundance is often measured using percentage cover or the biomass of the species. In bird communities, the abundance of the birds is measured by counting the individuals. And in fungal communities, for example, it is quite challenging to identify the abundance of the species based on the fruit bodies, and therefore we often record rather the presence absence of the species as fruit bodies. And this is important to remember, because this relates to how we will analyze our community data, as also will explain the different link functions and so on that there are for different types of community data. And here I wanted to make the point, which has already kind of appeared already in some of the questions that you have made, that all data come with errors. And there is quite little we can do about it. I mean, there is the perfect sampling method doesn't exist yet. All data will come with some sort of error, which we need to account for in our analysis, or at least acknowledge when interpreting the results. So one very common way, very common type of error is the so-called imperfect detection. So, for example, when in direct observational studies, when we go to the field and we directly record the species that we find, it's quite common to introduce false negatives into the data. This happens because the sampling effort that we apply is not enough to record all the species and individuals that there are in the sampling units. Even to the most expert people, this might happen, that we don't see some species that actually are there. This is called false negatives, and this is typical in direct surveys. False positives happen when errors like misidentifications introduce data on a species or individuals that are actually not present. So we record the species as present, but are actually not present. And this is typical in DNA-based surveys. In this kind of data that are now becoming more and more common. And this is something that we need to know. And then how we deal with it in our interpretations or analysis, then that is a step that we will need to take later on. And then another type of error that is quite common is the biased sampling effort. And this usually happens or is quite typical in data that originates from databases that compile many sources. So, for example, we may have in this kind of databases especially biased data toward where people live or the most accessible places, and we may also have taxonomic bias toward those species that are most charismatic or easiest to identify. So after this warning slide that we all need to think about what errors our data may contain and think about how to deal with it later on. Let's move on and let's introduce the environmental data. So the environmental data are those environmental data or coverage that we hypothesize to be important in determining the species that are in our community data. So this can be if we are recording non-manipulative observational data. This data can be, for example, the habitat type, some climatic conditions, and so on. And if we are working with experimental data, this is also the data matrix to include our experimental treatment. The environmental data, as you may have realized, it can be continuous or it can be categorical. If for example, we have climatic conditions, then this is a type of like precipitation or temperature. This is a type of continuous environmental data. And if we have the experimental treatment as a covariate, then we can include this as a categorical variable like treatment A and treatment B. And in the same way as with the species or community data, we can record this environmental data directly in the field or also from already existing databases. And here again, it's important to think if we are especially using existing databases, what kind of errors or biases this data may have and how that might influence the results that you are getting from your data. So usually the data have some spatial or temporal structure because we have different sampling units in different locations, and we may visit these sampling units several times. And then we have some data about this which we will later on treat as random effects, as you will see with Otso. So we have some data about the identity of the sampling units or the sites or the plots. We can have like some coordinate data, and in addition to this, we can have like the time in which we have done the sampling, and it's important to account for this in our analysis, as we will see, and as you most surely know, to account for the spatial-temporal structure of the data, so that we account for the temporal and spatial autocorrelation in our data. Quite often, we accompany our community data with some trait data, because we hypothesize that there are some traits that influence how the species are responding to the environment. For example, if we have two types of habitats with very different resources, we may want to include a trait that tells how the species uses the resources to understand the distributions of the species in the two habitats. In the same way as the environmental coverage, the traits can be continuous if we are, for example, using body mass or any kind of measure, or they can be categorical if we, for example, use some kind of the feeding type of our organism or color or whatever. The traits, we can measure them in the field. so that we measure the traits, we measure some characteristic of the species of the individuals that we record in the field, and then we make the species specific averages to include in our data matrix, because remember that we cannot yet include individual level data in HMSC, but we deal with species level data. But there are also some, they are becoming more and more common, these kind of trait databases in the literature and so on. And finally, when the aim is to study or to understand how the phylogenetic relationships affect the species responses to the environment, one can include phylogenetic data, a phylogenetic tree, for example. And the phylogenetic data is not usually measured directly, but compiled from existing databases that are becoming more and more common also. For example, the phylogeny of birds is very well resolved, and we have an excellent database to gather information about the phylogeny of birds. But for some other taxonomic groups, the taxonomy or the phylogeny might not be so well resolved, like is the case of fungi, for example. And in these kind of cases, we may use the taxonomic relationships of the species as a proxy of phylogeny, like what genus, order, class, blah, blah, the species belong to and how they are related. So now I have gone one by one through these data matrices that are typically collected in community ecology and which we can actually include as input in HMSC. So here I want to make the point that it's not necessary that you have all of these data matrix types when you want to run your HMSC analysis. The basic input data for an HMSC analysis is that you have your community data and the environmental data, so that you can link the responses of the species to the environment. This is a model which would have the community data and environment data. That would be like the most basic data matrices that I have explained. They are optional input data. You can include the community data, the environment data, and trait data, plus phylogeny or only phylogeny, plus the spatio-temporal context or not. So don't worry if you want to run HMSC and you don't have all these data matrices, because the very basic HMSC analysis already runs with a community data and environment data. And to summarize, in my lecture, I have explained how HMSC relates to theory. I have very briefly introduced how different HMSC parameters and outputs can be related to ecological theory. And I have introduced also what kind of data you can include in HMSC. So in the next lectures, you will spend most of the time understanding how to handle this data in your own computer, and hopefully you will be able to relate the patterns that arise from data to the ecological theory that I explained today. So Now I don't see my mouse here. So that you will spend the rest of the marathon in this part of this cycle, and hopefully we will be able to really relate patterns in data to theory as the ultimate objective. So thank you everybody for listening, and I don't know if we have now some time for questions or what is going on with the schedule. Mirka?
Sorry, I was scrolling questions.
Okay.
So we've been answering as fast as we can. One that just came in was about traits. So can you include site-specific trait data rather than species mean value? But I think not.
Sorry, can you the question again? Can you include like individual level?
Site-specific. So if you've got plots, so a plot-specific trait.
But then that you include it as an environmental data. When we are talking about traits in HMSC, we are talking about this kind of species-specific characteristics. And when we are talking about environmental data, we are talking about like plot or site or whichever unit characteristics.
Maybe one quick comment to follow up. So one of the graphic outputs of HMSC is actually a community weighted trait mean across the environmental gradients in the model.
Now you will in the next lectures you will see like what kind of outputs you get about the traits and the environmental covariates and so on. They were not explained at all in my lecture. So that you can get outputs like what are the predicted mean traits across different environmental gradients that you may have included in the model. But this will become clearer later on, I hope.
So there has been in the chat quite a bit discussion about these individual level traits or plot level traits, and as Nerea said, it's kind of not implemented yet that now the traits are at the species level, but I could mention this kind of a special trick or special case that you can do that, you know, let's assume that you are simultaneously interested in the presence absence of a species and then on some trait of the species, let's say how tall the trees are if you are modeling trees, and now the height of the trees is of interest at the plot level or whatever sample unit level. So what you can do in these kind of a cases is actually think about the trait of the species as a response variable. So you can place it into the Y matrix. You can have the same species twice in the Y matrix, once as presence absence, and then the second one would be the height of the tree conditional on presence. So NA is if it's not present, and if it's present, then it's the height of the tree. So in this way, you can kind of model individual or plot level variation in traits, but it's for a completely different purpose. Now it's a response variable rather than the explanatory variable, whereas in the mainstream HMSC application that Nerea was talking about, it's the explanatory variable more, and then it's only at the species level.
Yeah, and also, for example, if your sampling units are the trees, and then you are looking at whatever arthropods or whatever are in the trees, then as environmental covariates, you can include the characteristics of the trees, so that in that way you can also include these kind of tree individual specific traits into the analysis. But then you need to think a bit differently. So hopefully in the examples that we will see through the course, we will see like different examples that like these sort of things, they will become clearer and you will be exposed to these kind of different cases. So
there was a question about including traits as fixed effects, but that's not how HMSC works. I mean, is this too technical as well to discuss how traits feed into the?
Yeah.
I think this is best to leave to the part where I go through the equations and show how actually traits are implemented into the model.
Super. So are we ready for a lunch break, perhaps?
Yes, we are now 8 minutes before the schedule. So thanks very much, Nerea, for keeping up the time and having this great introduction to theory and data. So we will break out now for lunch, and we will be back in basically 1 hour and 8 minutes from now, as originally planned in the timetable. Okay, enjoy your breakfasts or lunches or dinners or snacks or whatever is the time where you are, and see you back in a bit more than an hour.